was asked to give just a brief, 10-minute presentation on the state of next-gen sequencing in terms of FDA regulatory review, and then open it to questions. So this will be a short talk on just, in general, how FDA regulatory review of clinical sequencing assays. To start with the disclaimer, these views are mine and do not necessarily reflect those of the US FDA or the US government. Particularly any funny bits or jokes I insert are not the US government's. So basically what I wanted to highlight is we do have recent clearances of NGS devices. We had an NGS platform that was cleared as a de novo 510K, and an NGS reagent kits that were also cleared as de novo 510Ks. We also had two assays that were cleared as class 2 in a traditional 510K. One is a variant panel assay, and one is a clinical sequencing assay. And these are what I'm going to use kind of as the case study, the example of how FDA regulatory review proceeded and the kinds of questions we asked. I'm not going to focus on the NGS platform and reagent kits other than to say the NGS platform was cleared as class 2 exempt with special controls, and the kit reagents were class 1 exempt. So if you're interested in this information can be found on our website where we have information on all the nucleic acid-based tests that we've cleared to date. And the decision summaries for both the reagents and the instrument platform can be found on this website. My slides will be made available after this talk. And finally, Giovanna Tizak within my office is going to give a talk specifically on the special controls associated with these clearances in the single-cell sequencing section tomorrow. So if you have lots of questions about instruments and reagents, that's the place to go. So now bringing my focus back to the assays actually serve as a great case study for how we thought about and the kind of information we asked for in association with the clearances. So in terms of the major elements of an IVD submission to the FDA, the intended use and the indications for use, if you don't know by now, is the key to how we review, the key to what we look at. And the review path, the types of studies, they all depend on the intended use claims independent of the technology or assay format. So absolutely give thought and consideration to the formation of your intended use statement. So here I've abbreviated the intended use statement for the Illumina MySeq Cystic Fibrosis 139 variant assay. It's a qualitative in vitro diagnostic system to simultaneously detect 139 clinically relevant CF disease-causing mutations and variants in the CFTR gene in genomic DNA isolated from peripheral whole blood specimens. It's intended for carrier screening in adults of reproductive age, confirmatory diagnostic testing of newborns and children, and as an initial test in the aid of diagnosis of individuals with suspected CF. Elements that are in the intended use include the Analytes. So there are 139 clinically relevant disease-causing mutations. The population that will be using the test, and the indication for use, in this case, carrier screening. However, the analyte can be a little tricky in NGS assays. In the variant panel assay, for example, it was a discrete number of variants. The variant types were well understood, SNVs, indels, et cetera. The variants had known clinical significance backed up with literature support, database support, et cetera. And the assay reporting was limited to the variants claimed. So the software output of that assay reported the presence or absence of these specific variants, not other variants within the CFTR gene. On the other hand, the clinical sequencing assay had a claim of the exon coding region of the CFTR gene. So this is a large number of possible variants, almost an infinite number of possible variants. The CFTR gene is humongous. So we did not have a discrete number of variants claimed. There were different types of variants possible in the sequencing assay. the types of variants that could be identified, different from reference, were of unknown clinical significance. Of course, there are variants of clinical significance, but you can't predict a priori what variants the assay is going to detect. And so, because of that, assay reporting requires trained interpretation. So the strength of NGS in this case can be clearly illustrated because there is a public health need to know about rare variants with known clinical significance but few samples. So we turn to the analytical and pre-analytical validation, which basically, in our terms, analytical validity is the assessment of, does my test measure the analyte I think it does correctly and reliably? In the case of the variant panel assay, where we had a discrete number of variants, the accuracy of the variants was established with clinical samples whenever possible. However, there is an acknowledgement that there were rare and extremely rare variants for which obtaining clinical samples was quite difficult. And so for those specific variants, accuracy was established using plasmid blends. The assay reproducibility was established using a panel of the variants with a subset of the panel. So in this case, the panel designed to assess the reproducibility of the assay has to be appropriate for the entire list. So it had to incorporate all the types of variants that would be seen in the variant assay in the genomic context of the types of variants it was detecting. On the other hand, if we look at the clinical sequencing assay, accuracy is claimed in the entire genomic region. So using clinical samples, we considered accuracy, which was based against Sanger sequencing of the entire CFTR gene for 500 samples. So that's all the bases, 500 samples. But once again, you can't have clinical samples for all the possible variants. That's impossible. So we'd have to consider accuracy both in terms of wild type and variant calls. The validation of possible types of variants claimed were incorporated into the review of the assay. So SNVs, indels, and we assessed what types of deletions or large insertions or deletions could be considered. So in this case, only the deletions they had specifically designed their assay to detect were able to be claimed. Other deletions could not be detected or claimed with this assay. And the validation of the assay performance in tricky regions. So CFTR has a homopolymeric stretch, and so there was considerable thought around the analytical validity of the test around this region. Likewise, in future submissions, we would consider analytical validity in homopolymeric, high GC content, other areas where sequencing might be tricky. In terms of clinical validity, so clinical validity in our terms is, does my test result correlate with the expected clinical presentation and how reliably? So this is the phenotype-genotype link. So for the variant panel assay, clinical validity was already established for the cleared variants. The predicate device for this clearance was the Luminex CF60 assay. So for those 60 variants, clinical validity is already established. Clinical validity for the new variants was actually established using the CFTR2 database, which is a well-documented and curated database. So in the future, when designing these kinds of assays, you should consider the source of clinical validity. And I absolutely encourage you to engage with the FDA to discuss whether or not this source of clinical validity is appropriate for a submission. Now, on the other hand, the clinical sequencing assay has no clear-cut clinical validity because it's reporting all differences from reference. It is well known that most of those will be benign polymorphisms, SNPs, meaningless, not meaningless, but have no clinical phenotype. So the assay reports variants from reference with no annotations on significance and puts a large burden on assay interpretation. So here, the strength of NGS is that there is a public health need to gather data and understand the genotype-phenotype link in complex disease. And this is an excellent way to gather that sort of information. However, it draws attention to the fact that it's shifting the burden of interpreting the assay into the hands of the end user. And as such, the assay complexity is deemed high, and the labeling is written to reflect that. So moving on to the labeling. In the variant panel assay, labeling to validate with a second method, we incorporate labeling to validate with a second method variants that were analytically validated only in plasmid. So as I said, some of the rare variants we allowed analytical validation with plasmid, but then we mitigated that risk in labeling by telling labs that these had not been analytically validated in a clinical sample. On the other hand, the clinical sequencing assay has labeling that the test is most appropriate when the patient has an atypical or non-classic presentation of CF, or when other mutation panels have failed to identify both causative mutations. And the results of this test are intended to be interpreted by a board-certified clinical molecular geneticist or equivalent. So what we are saying is that the labeling has been written to kind of mitigate the risk of the fact that just because you have an output of a difference from reference doesn't mean there is clinical validity and does not mean there is a genotype, phenotype, or pathological link. So in conclusion, I want to emphasize that the review path, the types of studies, everything depends on the intended use claims. And you should think carefully about what you want your test to detect and how you want your test to be used. Establishing analytical and clinical validity creates unique challenges for NGS, but they are surmountable, and I absolutely recommend coming to us early.
Okay.
And the recent clearances can serve as a case study for future NGS-based assays, and early interaction with the agency is extremely valuable. The pre-submission process is free. We highly recommend you utilize it, and customer service is very important to us. So thank you. I'm available for questions here, but my email address is there, and I'd be happy to take questions over email.
My hat's off on the clearance of the instrument as well as the assays last year. That was a big milestone. And
yes.
Interesting when you look at both sides of those tables, how different it is from what we've known as a historical IVD.
Right. I think historical IVDs didn't actually have the option, like the clinical sequencing side, without an established clinical validity.
Looking at the area of oncology, because CF, I think we have a lot of examples of sequencing in CF and what we've done in that way. I mean, it's not anything, I don't want to say it's not anything new, but we've been doing it for a while.
Yes.
Looking in the area of oncology, and so thinking about, can we have a multi-gene, multi-transcript panel be an approved diagnostic platform?
Yes. So we don't have one yet. So there is no cleared multi-gene, multi-transcript platform as of yet. However, I think I drew these as case studies because I think this is an example of the sort of things, especially in oncology, people need to think about early when crafting their device design and their intended use statement. Is the intention to select patients? And if the intention is to select patients for a therapy, it behooves you to use clinically valid variants to do this, right? So if you are using well-established clinically valid variants, then making a panel assay of these is probably the most appropriate way.
Okay, so we know that in this world, every time I turn on my PC, there is a new variant of sorts. So
Yes.
This panel will never, or maybe will, I don't want to say never, but might never be static. So how would, if I introduce a panel of 50 genes, 100,
Right.
pick it,
Yeah.
how would I then start to think about updating that with the FDA on a regular basis?
So it depends on how it's been cleared or approved. And this is actually, this is a policy that's still undergoing discussions, because like I said, we don't have a cleared or approved multi-gene, multi-transcript panel yet. However, what I would encourage you, it would depend on whether it's a PMA or a 510K. It would depend on the extent of change that you're doing, and it would depend on the output of the assay and what the extent of the changes are, the data you have to support these changes, and the reasons for these changes.
Mm-hmm.
I don't know if I can give more details than that.
Well, we'd love more detail if that's possible. But I get the feeling that based on what you're doing, it's going to be a big shift from anything we've ever done before on this front.
I think we've acknowledged the need to be flexible. However, we've acknowledged the need to be flexible, and we're a naturally cautious organization. But I think in terms of making changes or adding to a variant panel or changing how to interpret the results of a variant panel, these all depend on the design of the assay and the data you have to support those changes.
So I guess that brings up another part. I mean, I see three parts of regulation here. One is obviously the instrument.
Right.
The next one is the assay.
Yes.
But then you keep using the word interpretation, and I'm wondering at some point there's a piece of software there.
Yes.
That is called clinical decision support.
Which
if I'm not mistaken, the Congress has said that should not be regulated.
So interpretation of the assay all depends on the assay output and the level of complexity. Like going back to use these as an example, in the variant panel assay, it reports the presence of that specific variant. So the interpretation is not complex, right? The software associated with the instrument isn't clinical decision support software. It's software that outputs the results.
Correct.
Likewise, in the clinical sequencing assay, all it reports is difference from reference, no interpretation, right? So once again, that's not clinical decision support software. That's shifting responsibility to the medical professionals, or in this case, the molecular geneticists, to interpret appropriately based on the current state of the literature, the current state of knowledge, the current state of, and the clinical covariance associated with that particular patient sample. So I wouldn't call this kind of output clinical decision support. Now, say someone wanted to create an assay that you put in a sample and it put out a diagnosis, like this would be a higher risk proposition with a different level of validation that would be required. Do you see how it's different?
No, I know, but I'm trying to project where some of these assays may be going in the future.
Right.
And the future is not that far away.
Yep, it's tomorrow. But yeah, so our policies are evolving, and I think always we look at it in terms of risk benefit, right? And we look at it in terms of the risk of getting a patient an improper procedure, making an inappropriate diagnosis, which would expose them to dangerous, potentially toxic drugs. And so when looking at it, it depends the indication, it depends on the risks associated, and it depends on the amount of data or the comfort in the field, because we've had tremendous advances. We know so many more variants that are very meaningful, but we've also taken steps back. We've also gone, oops, that one's not pathogenic really. And we have to understand that this is a young science.
Or it's pathogenic and it's not on.
Well, or it's pathogenic with very low penetrance. Yeah. We have a lot to understand. And so I think that's why early interaction with the agency and early understanding of the risk associated with making a change to your panel should be understood best by you, right? And you should be able to come to the agency with like a reasoned scientific justification for whatever kind of assay output you want to do. But I don't think in the case of the clinical sequencing assay, where it just outputs difference from reference. That's, that's putting a large burden on the lab to interpret the results. But at this point, like, that might be a good place to be.
Correct. Yeah. No. I mean, I think some people are going to start there, but I've already know of, or I'm talking to people that are talking about where they're going to be in the next 3 years or 4 years, right?
Absolutely.
And the agency has to be prepared for those sorts of changes.
Absolutely. And I would say come with data and information.
Once you approve these existing sequencing-based assays, now the agency has blessed it, cleared it, et cetera.
Yes.
Does the agency have a position as that? How does that compare to somebody who's, for lack of a better term, homebrewing it in their own lab versus one that's approved?
Okay. So as we all know, the FDA has been working on a regulatory framework for laboratory developed tests for many years. However, today, at the current state of our development of this regulatory framework, I cannot comment. So I'm so sorry if you came to hear me expound on LDT regulation, you won't get it. I'm going to say no comment in regards. I can refer you to our office director, Alberto Gutierrez, and I can also refer you to Elizabeth Mansfield. Both of these people are able to answer questions on laboratory developed tests at this time, but I am not. So I'm so sorry to disappoint, but I cannot at this time talk about laboratory developed tests.
Damn.
I know.
I tried, okay.
He tried. He tried really hard.
We even talked about it ahead of time. So does the indication that the FDA is now moving forward and clearing certain assays on the NGS side, is that a predictor of how it's looking at other technologies like digital PCR and those sorts of things?
Well, I think there's been a lot of technology advancements. I think the digital PCR systems are becoming more mature. I would anticipate them coming to the agency at some time in the near future. I think fundamentally our NGS kind of policies can be translated to other technologies. But yeah, they can't necessarily be used as a predicate in the classic regulatory sense. They would have to come in with an assay and then depending on the indication. But I think the newer multiplex technologies will benefit from our work on the NGS clearances.
So let me go back to, you had said something about the CF test and then how Sanger sequencing was introduced. And so, but Sanger and NGS are different beasts.
Yes.
So how do you reconcile concordance between, because in reality, you're not really comparing apples and apples.
Right. So in terms of accuracy, and this is where we get to analytical validity as opposed to clinical validity. And in terms of analytical validity, you have to establish that the variant you detected is there and you haven't missed a variant, or it's there reliably and reproducibly. So for an accuracy standard, you have to compare to something else. In the case of the cystic fibrosis assays, they compared to the sequencing using Sanger. Interaction with the agency is highly recommended. You can compare with other NGS platforms. You can compare with known, well-established known sequences. You can compare in a number of ways to generate your accuracy standard. But basically, you would compare your technology to truth, established through a reference method, established in some analytically valid way.
So that being said, will there come a point, how many of these do we have to get cleared before we're not comparing it to what some people might say is an outdated standard?
So right now you can compare to something other than Sanger. So we have one cleared. So, but devising your accuracy study for your analytical validation, you can use something other than Sanger. Frequently, we default to Sanger because Sanger is very well established. It's been frequently used as a reference method. It's frequently used as the reflex testing in clinical labs using NGS. And so it's the simplest, most obvious, easiest kind of analytical reference standard. However, if you have a strong case or some reason that you want to use an alternative method for your analytical reference standard, we certainly entertain that. We aren't saying it has to be Sanger, but I'm saying it's typically Sanger, and Sanger has advantages in that it's frequently already the reflex test for accuracy testing in NGS.
So I want to step back one step to the interpretive piece of this, because I do believe that that's going to come to a head in the next 12 to 24 months at some point. How is the agency thinking about software that, I don't want to say makes the interpretation, but participates, if you must be, in the interpretation of what's being output?
Okay, so any software associated with this assay system, we'll call it a system, because in the end, what's being cleared is a system. It's the input, it's the instrument, it's the output to the final paper, the product. So in the review of an assay system, that software would be incorporated. So when you get from your FASTQ files, your VCF files, et cetera, to whatever your output is. And now your output could be, difference from reference. Your output could be raw sequence. Your output could be diagnosis of CF. Now, these three different outputs have three different risks associated with them, right? Because in one, you don't have the chance, the output is simply black box, no sequence, just diagnosis of CF. You see how that's higher risk because you can't go back and manually confirm that. Now, if it's raw sequence and you're just allowing, it's not very useful, but it's not very risky. Now, which is why I think people are tending to lean variant from reference as their output. But we consider the whole system in the review. And so from input of the sample, pre-analytical preparation requirements, to the outputs, all of that is part of the assay system and would be incorporated in the clearance or approval.
Interesting, because I'm noticing the institutions actually putting together their own software.
Right.
To help with that next piece. So it's different than the company producing the assay.
Right, right. And that's something we've been considering. But if the assay manufacturer output is X, and then they're leaving it to the end user to do with that data what they will, that's a different question. And an evolving policy. I think it's an evolving policy within the agency and within the clinical labs. What softwares do you use? Do you trust them? How many verifications do you need? Most of the people I talk to use multiple software packages, don't rely on just one, and reconfirm things. And I think there's a need for good databases. I think there's a need for well-curated systems and help with making clinical decision making, and I think that's a discussion that the agency is having with the outside community very actively.